طيب, Assalamu alaikum everyone. I am Talah Nammor, a medical intern, and today we're going to touch base on everything, inshallah, you need for the OSPE. I know POD is, let's not say hard, let's just say a bit challenging, but I'm pretty sure you guys got it. So, yalla, let's start. The way that I did it, it's not, it's questions based. It's nothing like, it's not يعني I'm not here to teach you guys, I'm here to test you, okay? And revise with you. فيلا, let's start. So, يلا, who wants to read? Can someone read the question for me? Guys, I need you to be يعني interactive معي, هيك ما بزبط. If you're not gonna read it, I'll read it. But I want you guys to read it. Can someone please read it? Oh, okay, fine, I'll read it. So, a 55-year-old diabetic male admitted to admitted after an emergency cholecystectomy. So cholecystectomy is just a surgery, no need to know the details. So on post-op day 3, he developed fever, hypotension, and purulent discharge. All of this came from the surgical wound, okay? So a post-swap was taken and we saw this beautiful image. You guys don't need to feel scared because of the scenario. The scenario is just there to, يعني start the show, okay? So what do you guys see? Describe what you see in this photo. Guys, I need you to be interactive. Tell me, what do you see? Let's start with color. What is the color that we see? Okay, gram positive. Why are we saying gram positive? لأنه it's purple, right? Because gram positive is purple and negative is red. Diplococci gram positive. Okay. Why did you decide it's diplococci? ليش ما نحنا let's take it step by step قبل ما نروح ننط على diplococci. It's cocci. So what is a cocci? It's basically circle in shape. It's a circle. تمام? And if you look at it clearly, basically we can't call it diplococci because diplococci is only two. But هون we can see مثلا, like look at here, we have a bunch of 3 or a lot of this. So this is clusters. So we have gram-positive cocci in clusters. Okay? Okay, good. Yalla, let's go on. So what do you think the genus is? So this is most probably a staph. Okay, okay, I like. So we probably think it's a staph. Okay? تمام. طيب. Let's say this is a question about stains, that I used a negative stain. So if I use a negative stain, the color will appear where? بالبكتيريا ولا بالbackground? Background. Okay. يلا. Do you all agree? Perfect. So when we use a positive stain, usually it clicks to the bacteria, but a negative stain will go into the background. حلو. طيب, give me an example of a negative stain. I just want to make sure that you guys Zeal Nielsen. في one that I'm not sure about Zeal Nielsen بصراحة. But there is one that is really like I don't think Zeal Nielsen is a negative stain صراحة. But there is one that is very like هيك unique. Yeah, it's not exactly a negative stain, Zeal Nielsen. So basically, I'll make it easier for you guys. India ink. Have you heard of India ink? So India ink is an example for a negative stain. So if they use India ink, just know that the color will appear in the background, not in the bacteria itself. Is that clear? Okay. طيب, this is an extra bonus question. It's a trick question. I don't know if you guys are gonna know it, maybe لا, but let's see. Do you know what organism India ink is used for? Or like, where does it help? Fungi? Okay. So, okay, this is an extra information. Don't stress about it. خلاص, just forget about it. But it's basically Cryptococcus neoformans. It's basically like an organism that causes like things in, yeah, exactly. So it causes meningitis in immunocompromised patients. Okay? This is an extra information. I'm not sure if she touched upon it يعني, but I just wanted to see if you guys know what India ink is. بس ما شاء الله you guys are doing great. طيب يلا, what are the differences between those two mediums? So we have blood agar and MSA. So all of these are questions they might ask you. It might be written or it might be MCQ. So يلا. Okay. MSA is both differential and selective. I like. What about blood agar? Only differential. Exactly. So blood agar is only differential and it's enriched. What do we mean by enriched? That it has nutrients and specific like let's say chemicals that help specific bacteria, or not specific only bacteria, organisms live on it. Okay? So it's enriched and differential. So it helps us differentiate between the organisms that grow on it. تمام? While on the other hand, MSA is selective and differential. And why is it selective? Can anyone tell me why is it selective? What does it contain? What's the thing that it contains that helps us know that it's selective? Mannitol, exactly. So any bacteria that is capable of fermenting mannitol, as you guys can see, we have a red side here and a yellow side. So the red side, basically bacteria that cannot ferment mannitol are going to grow there. But if they do ferment mannitol, the red is going to change into yellow. And that's how we know it's a differential. All right? And it's selective. It has both. Is that clear? Okay, perfect. So we're going slowly. So this is just like a very fast review of the different media. I'm not gonna go into details يعني about each and every one. Just I need you guys to know what is the the things that they have. So مثلا blood agar, chocolate agar, they're enriched media. McConkey, mannitol salt agar, Lowenstein Jensen media, all of these are selective. And I want you to understand, what does enriched and selective mean? So enriched means it helps specific organisms grow in it. Selective means it helps us know which one is going to stay and which one won't, all right? We have indicator at differential media. It helps us to differentiate between organisms. Transport media specifically to help us transport the organism. So let's say we don't have the organism needed in a certain lab and we want to send it to another lab. We usually use transport media. Storage media is something just to store in it. Okay, let's move on. So, okay. Remember the case that we started with, the gram-positive cocci in clusters? So this organism was cultured on this media. So name the mediums now again. We know what is this. This is MSA and this is blood agar, sorry. This is blood agar. طيب, what are the changes that we see? There's a change in color from red to yellow. This means it's a mannitol fermenting organism, all right? And in here, if you could look, it could looks like pearls, or it looks like, I don't know how to explain it. It looks like, you see how white they are? Okay, beta hemolytic. Let's not go there yet. Let's wait. I just need you to know that's mannitol fermenting, and what is it called? It is beta hemolytic. Yeah, it is. Okay. So let's move on. Then another test was done. So now we know it's stuff, right? So we want to change to kind of figure out what kind of stuff is it. تمام? So we do this test. What is the name of this test? Catalase, exactly. So basically, we have an organism. So we put basically the organism in here, and then we add on it reagent to see whether is it catalase positive or catalase negative. So if it was catalase negative, it's not going to have any reaction. But if it was catalase positive, we're going to see a bunch of bubbles. And these bubbles signify that this organism is catalase positive, all right? And in here, the organism is catalase positive because it showed us this. All right, let's move on. Then we did another test. So what is this test? Coagulase, exactly. So coagulase is another test that can help us differentiate between the different stuff that we have, okay? So coagulase, in this, let's look at this beaker. So in this beaker, what do you guys see? Nothing, exactly, we see nothing. So then we add to it the reagent and then this happens. And this is like literally a blood clot. So we have clotting. So coagulase, all right? Coagulase, exactly, coagulase, if the bacteria that we have is coagulase positive, it's going to result in coagulation. And because of coagulation, we're going to see this clot. So let's keep in mind. So we have an gram-positive cocci in clusters that is catalase positive and coagulase positive. Do you have any suggestions what the microorganism might be? Staph aureus, perfect. طيب, do we have one more test that if we do, خلاص, we know 100 million% that it's Staph aureus? Do you know the name of the test? No. There is a third test, بس يعني this third test confirms إنه Staph aureus because it's positive in all of these tests. Do you get what I mean? So like coagulase, catalase, and what's the third one? Okay. Third one is the DNAs, okay? So this test is basically we put the microorganism and we check if there will be a change in color and if there will be, you see this area of whiteness around it. So this means that it has DNAs which will break down the DNA that is found in the culture. Okay? That is found in the medium يعني. For this will signify إنه it does have DNAs, which basically only Staph aureus has all three, catalase, coagulase, and DNAs. Is that clear? To everyone? Okay, if you have any questions, stop me at any time. Okay, so this is what we were just discussing. So basically, gram-positive cocci, we do the catalase test. If it's positive, then we know it's a staph species, all right? If it was negative, then we would think of streptococcus, all right? طيب, it was catalase positive. How can I differentiate between the staph species? We do the coagulase test. Now, coagulase test, if it was positive, then it's Staph aureus, not Staph epidermidis. If it was negative, it's Staph epidermidis. If I want to be 100 million, يعني 100% sure that it is Staph aureus, I could just do DNA tests, like the DNAs, and if it was positive, then it's 100% Staphylococcus aureus. Is that clear? Okay. Now, while we were basically processing another ICU patient, the same lab was able to isolate this picture. So يلا, what do you guys see? First of all, it's red, so it is gram-negative bacilli. Exactly. So they are rods and they're red, so gram-negative bacilli. Perfect. Now let's look at the McConkey. What do you guys see on the McConkey agar? Lactose fermentation, exactly. So for reference, this is the McConkey, all right? And then if you look at these like changes on the top, this is lactose fermentation. Now, we have multiple microorganisms that can cause lactose fermentation, but there are differences between the like if you look at it and try to describe it, there are different between the characteristics of the lactose fermentation. So in E. coli, look at how these are non-mucoid, like they're just dots. They're just dots at the McConkey, like they're not mucoid like the Klebsiella. Like if you look at the Klebsiella, you could see how it looks slimy. Is that clear? So let's say now I'm trying to like cover a bunch of concepts in the same idea, you get me? So in here we're talking about McConkey. McConkey, what does McConkey help us do? It helps us understand if an organism is lactose fermenter or non-lactose fermenter. Okay, so if it does have these dots all over it, then it is a lactose fermenter. طيب, how can I differentiate from the way the lactose, like the morphology of the lactose fermentation? لأنه we have a lot of microorganisms that can be lactose fermenters. So from the morphology only, like if I'm only looking at the plate, if it was non-mucoid, I can think of E. coli. If it was mucoid, I think of Klebsiella pneumonia. All right? Now let's move on. So this test was done on the ICU patient. So does anyone know what this test is called? Oxidase test, exactly. So basically, the microorganism was put there, and then we added a reagent, and then we tried to see if there's a change in color. And in here, there was a change in color. It changed into purple. This indicates that if we see this purple stripe, that it is oxidase positive. All right? Okay. Now, there's another test. So this is the normal reagent, and then this is after. So what is the name of this test? No. Um, yeah, indole صح. It's another name for the urease test. Okay? So basically, the indole test or the urease test, what happens is that when we add it to this, like when we add the microorganism into this beautiful medium, it's going to change its color into pink. So let's think together. This organism is a gram-negative bacilli. It is oxidase positive. It is urease positive. So what is the first microorganism? And it's a lactose fermenter. So what's the first organism that comes in mind? E. coli. Is E. coli, what is it called? Uh, Oxidase and urease positive? You're not answering my question, is it? Klebsiella. What about the rest? Exactly, it is Klebsiella, so the rest is not saying anything. طيب. So an organism that is oxidase and urease positive and is a lactose fermenter, we have to think of Klebsiella. تمام? Is that clear? Okay. Now, another test. What is this? Gram-negative bacteria, صح? Gram-negative, specifically bacilli. Now, let's look at the McConkey. The McConkey is not... What do you see on the McConkey in here? There is no lactose fermentation. Like, this is just the strike of the like culture. طيب. And it is oxidase negative. And it is urease positive. What are you thinking of? So non-lactose fermenter, urease positive, oxidase negative. And I think I have like a, wait. Yeah. And this is a trick, come on. Like not a trick, Blacks. This is something to help you, a hint. So swarming motion in the agar. What are we thinking of? Proteus, exactly. So so if we see gram negative, so we have a patient that has like ICU, that is in the ICU, has whatever symptoms, I don't care about the symptoms, all right? Has gram negative bacilli with no lactose fermentation, with urease, oxidase negative, urease positive, and a swarming culture, just think of Proteus. Is it like hard? Or is it confusing? Yeah, okay, fair. I just don't want you guys to feel confused. All right, let's move on. So basically, do you guys know what is this test? So this test is basically a test that they use to know sensitivity. So what do? We get the organism, and then we get مثلا 3 different antibiotics. We plate them with the microorganism. Let's say this in here. The whole مثلا this culture has Staph aureus, let's say, all right? And then we have 3 different antibacterial, and then we leave it to grow, and we go and check basically the zone around these like small pills, just to know how much is there. And what do I mean by how much is there? So basically you see these lines in here. This is bacterial growth. So this is a place where we know bacteria is there. So مثلا, let's look at pill A. Around it, we don't have any white area, or around it we have like a lot of bacteria. So this means that A has no effect on the bacteria. Let's look at B. There is a white zone around it. And what does this white round zone mean? This means that the bacteria was not able to grow around this region because of the activity of the pill. Now let's look at C. Come on, we have a lot of white area. So which in this photo مثلا, which one is the best to choose out of these 3 شو اسمه antibiotics? Obviously the answer is going to be C, because it has the biggest radius. تمام? Okay, so what does AST measure in principle? Again, we talked about it, the in vitro growth inhibition. So we're trying to check how much is this medication preventing the growth of the bacteria. تمام? طيب, هلا let's say this in here, okay, is methicillin. طيب, so if methicillin is here, and then there was bacterial growth around it, and this is stuff, this, we are culturing stuff. So what is the name of this stuff that is resistant to methicillin? MRSA, okay? طيب, question that has nothing to do with OSPE. How do you treat MRSA? Like, what's the first antibiotic that they usually use? Vancomycin. Perfect, guys. طيب. Is everything clear up until now? Okay, perfect. طيب يلا, let's take another case. So a 9-year-old child presents with sore throat, fever, and enlarged cervical lymph nodes. The tonsils are erythematous with grayish-white exudates. A throat swab culture was taken, and it was even cultured on the blood agar. So yalla, let's look together. What do you guys see in here? Gram positive because it's purple. Is this bacilli or rods? What do you guys think? Okay, gram positive cocci in chains. Perfect. So we have gram positive cocci in chains. All right. Now let's look at this. Do you remember this? This was the catalase test. So catalase test, we don't see any bubbles. So this is catalase negative. All right. And let's look at this disc. All right. So we haven't discussed hemolysis yet. So we have this concept, which is basically we're going to have blood agar. Hello? Now, the blood agar, from the name, it's going to contain blood. Tamam? Certain microorganisms or certain bacteria, they have the ability to hemolyze the blood. And by hemolyze the blood, it means break it down. So when they break it down, it becomes clear. All right? And we have three kinds of different hemolysis. So we have alpha hemolysis, beta hemolysis, and gamma. So alpha, basically, it's partial hemolysis. It's not complete hemolysis. They don't have all the guns and things they need to completely hemolyze the agar. Beta hemolysis, basically, the agar would be clear because they're able to hemolyze the blood completely. And then gamma hemolysis is basically when they do not have anything that would help them to basically hemolyze the blood. So in here, do you guys think this is alpha, beta, or gamma? Beta hemolytic, exactly. Because you can see that some regions are, يعني, you can't see any blood in it. So they're completely hemolyzed, all right? So, yalla, beta hemolytic, gram positive, cocci in clusters, and it's catalase negative. So what are we thinking of? هلا قبل ما أصلا نحكي عن beta hemolytic, catalase negative means it's a streptococci. حلو? So we're talking about a streptococci. Great. This streptococci is not alpha ولا gamma, so it's beta hemolytic. So we have two options in mind. What are the two options that you guys have in mind to know what organism is this? Strep pyogenes is one, perfect. What are other beta hemolytic bacteria? كمان في another one, a galactia. Exactly. So هلا نحن if we wanna differentiate between Streptococcus pyogenes and Streptococcus agalactia, what are we going to do? We're going to do a sensitivity test. And what is the pill or the medication that we're going to try? يلا يا شاطرين. Bacitracin, perfect. So let's look at this case in here. So bacitracin was tested and it showed the following. So this is bacitracin. And in here, do we have a zone of clearance or no? Or a zone of inhibition? Yes. So what are we thinking of? Is it strep pyogenes ولا agalactia? Pyogenes, perfect. Tamam. So you guys are getting the idea. This is all we need to know. So strep pyogenes is a gram-positive cocci that is in clusters, catalase negative, beta hemolytic, and it basically is sensitive to bacitracin. هلا, if it was not sensitive to bacitracin, if it was resistant and we had growth in here, then we would think of strep agalactia. تمام? Okay, I think خلاص we mentioned these. So what is the organism? Strep pyogenes. The hemolysis was beta hemolytic. What test was done that it differentiated from group B streptococcus? It's not the catalase. Ignore that. It's not the catalase. It's basically bacitracin bacitracin sensitivity, and these are 2 other complications. This is basically all the things that strep pyogenes can cause. It can cause pharyngitis, scarlet fever, erysipelas, or pyoderma, okay? These are things they're not really important in OSPE. تمام؟ يلا هلا نحنا we talked about alpha hemolysis, beta hemolysis, and gamma. So do you guys see the difference? In alpha hemolysis يعني it's not clear because it did not fully hemolyze the blood, but it's dark because it kind of did a reaction, okay? Beta hemolysis is خلاص, it's clear. There is no any color. And then gamma, it's just like you drew with on the on the agar because it does nothing. تمام? Clear? So this is basically how we can differentiate the cocci. So gram-positive cocci, it can either be negative or positive. How do we know if it's negative or positive? By the catalase test. If it was catalase positive, then we do staph. We think of staph. If it was catalase negative, we think of strep. Okay, خلاص, we know it's strep. How do we know and know what? We have so many different kinds of streptococcus يعني. So how do we know? How do we differentiate between them? We do the hemolysis test. So if it was beta hemolytic, we would think of strep pyogenes or strep agalactia. If it was alpha hemolytic, we would think of strep pneumonia or strep viridans, okay? So how do we differentiate between strep agalactia and strep pyogenes? Bacitracin. If it was sensitive, then it's pyogenes. If it was resistant, then it's agalactia. And then for the alpha hemolytic, on the other hand, okay, the alpha hemolytic, لا wait, قبل ما نروح الألفا. Let's go to the alpha hemolytic. So for the alpha hemolytic, we have 2 options. It's either going to be strep pneumo or strep viridans. صح? Streptococcus pneumonia, we use another kind of medication to check for sensitivity. And what is this medication? Opticin. Perfect. Opticin. So we have opticin resistant and opticin sensitive. What do you think is sensitive to opticin? Which one of these? Strep pneumo or strep viridans? Pneumo, exactly. So if we put the disk of opticin on it and it showed a zone of inhibition, then it's pneumonia. If it did not, then it's strep viridans. And I don't know if you guys شو اسمه strep. I don't know if you guys watched the sketchy for it. Like it's so like I watched that sketchy 4 years ago and I still remember how like the viridans and the pneumonia, they're like fighting and stuff. Yeah, it's really amazing. No, for fact. This is how we can differentiate between different streptococci حتى الستaphylococcus. We talked about it before. Is it clear up until now? Because this is what يعني they're not gonna tell you. They might just show you the organism. They might just show you a test they did and they're like يلا identify this microorganism. تمام. هلا السترeptococcus agalactia, okay, has an extra test that can help us to know that it's Streptococcus agalactia, which is basically the CAMP test. Now, what is the CAMP test? The CAMP test, it's just, you see here how basically okay, let's explain it. In the CAMP test, what they do, this is a culture of Staphylococcus aureus, okay? Why do they culture it? Don't ask me. I don't know. They just literally they culture it, okay? And then in here, they're going to put, let's say, Streptococcus agalactia, and in here, they're going to put Streptococcus pyogenes. Now, what they are looking for, they're looking for this triangle formation. Now, if Streptococcus agalactia, if the microorganism that is cultured in here and in here it's Streptococcus agalactia, if it causes this triangle to be formed, which is basically just like you can think of it as a way, and Streptococcus agalactia loves Staphylococcus aureus, and if it sees it, it's going to this is just its way of welcoming it, which is the arrow. If this happens, then it's CAMP positive, then we know for a fact this is Streptococcus agalactia. But if the triangle did not happen and it's just like a normal line, there is no this huge welcome, then it's probably any other kind, which probably is Streptococcus pyogenes. Is that clear? Is the camp test clear? So if the camp test is positive, we think of Streptococcus agalactia. So يعني it's like an additional exam. Sometimes so basically extra hemolysis, exactly. So when extra hemolysis happens when it meets Staph aureus, this is Streptococcus agalactia. And this is just a test that further proves إنه it's Streptococcus agalactia. يعني إحنا if we did the bacitracin test and it was resistant, then you know for a fact that it's Streptococcus agalactia. And if you want to know, like, يعني you want to just make sure that whatever your choice, your choice was right, you could just do the CAMP test and you can see this huge hemolysis or like this extra welcoming. And what is it called? Sometimes they might just show you this camp test, give you a little bit of, give you a small scenario and tell you what do you think it is, and you just have to know that camp positive is Streptococcus agalactia, because I don't think any other microorganism in this lecture that was, or in your material that was mentioned is camp positive. Come on. Is that clear? Okay, good. Now in here, this is the alpha hemolytic. So alpha hemolytic, we said, it's going to change color, but it's not going to fully be transparent. What are we thinking of? We're thinking of strep pneumo and strep viridans. Now this is optogen and zone of inhibition. Then we think of strep pneumonia. Okay. In here, come on, this is just a repetition. This is strep pneumonia and this is strep viridans. Clear? So optogen resistant, strep viridans. Optogen susceptible or sensitive, strep pneumo. Moving on. So this is a quick differentiation mnemonic. A is almost, B is best clear zone, and Y is gone hemolysis, يعني ما في hemolysis. And the gone hemolysis is usually enterococcus, because we're talking here about cocci. We're talking about the circles, all right? This is just a table that I did for you that you guys can use to just revise really, really fast. No need to يعني memorize it, but just know it إنه like مثلا strep pneumo, alpha hemolytic, it's catalase, all of them are catalase negative obviously. Optogen sensitive, it's biosoluble. Viridans نفس الشيء بس optogen resistant and not biosoluble. حتى the biosolubility كمان was mentioned in sketchy video. Then عنا البeta hemolytic, the clear, the complete. Bacitracin sensitive, strep pyogenes. Bacitracin resistant, strep agalactia. CAMP test positive, strep agalactia. يعني just guys في هيك أشياء بتحطوها ببالكم and you directly know إنه oh this is that. هلا if you guys look at this slide, would you يعني what's the first thing that has to come through your mind? Knowing that, before I complete, knowing that it's a cocci. It's a gram-positive cocci. Exactly, enterococcus. So enterococcus, you can see this black thing. It grows in basically 6.5 sodium chloride. تمام? Is everything up until now clear? حلو. A 32-year-old male from a low-income setting presents with chronic cough, low-grade fever, night sweats, 5 kilogram weight loss. يعني صراحة أنتم من الكلينيكال بنات لازم حالكم تعرفوا إنه this is TB before you guys see this. But yeah, blah, 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 blah. I don't care. We look at the thing that they showed us. What is the stain used in here? Acid-fast stain. Ziehl-Neelsen stain. Okay? Okay. What is this basically? Do we see anything in the Ziehl-Neelsen stain? Yeah, we see basically TB. طيب. Name the primary dye and the counter stain used. يلا. Don't tell me these details are not important. They are. Because I don't know, while doing these slides, صراحة, guys, I remembered a couple of my POT questions, and I think I got really stringent questions. So do you guys know what is the primary? So في عنا primary dye, then عنا counter stain. The primary dye is basically the pink one, which is the carbofuscin. أنا حافظة pink إنه بس إنه فوشيا يعني هيك in my mind carpal fusion is fuchsia. It might not be the actual pink thing بس يعني. والcounter stain is the methylene blue. حلو؟ هلا we need before ما we go through the steps. يلا, why do acid fast bacteria retain the primary dye after decolorization? What do they have? Because you know how it is. We give them the primary dye, we kind of heat them, and then we rinse them, then we give them the counter stain dye. طب ليش acid fast bacteria retain this? Can anyone tell me? Yalla guys, it's the thing that basically, sorry, it's the thing that differentiates like this bacteria from other bacteria. Something in the cell wall. Cholesterol, yeah, close enough. Mycolic acid. So the presence of mycolic acid, okay, in their cell wall is the thing that will retain initial is the thing that will contain initial, يعني it's the thing that will preserve the initial color. Do you get what I mean? Is that clear? Okay, nice. Now, this is important. اللي هي steps of what is it called? I remember, I don't know if my mind is playing games on me. So I remember they asked us in an SAQ أو حتى OSPE to write all the steps of this. فاحفظوهم. So basically, it's easy. First step, you apply a primary stain اللي هي carbon fusion. Then you heat it. And then you rinse it by basically an acid alcohol. So you kind of try to decolorize it. And then you add the counter stain. And then you look at it. This is how you see. So basically, لأنه عندنا mycolic acid in TB or like a specific acid fast bacteria, because they have TB, they have mycolic acid, they will retain a carbon fusion, and that's why they look pink. So you see these pinkish like clusters, these are the bacteria. Okay, I hope it's clear. هلا types of staining, we have multiple types of staining. We have simple, we have differential, we have special. هلا simple staining positive و negative. إن negative عنا India ink اللي حكينا عنها. بالنيجاتيف الباكجراوند هي اللي background is the thing that is basically dyed. Differential staining عنا gram staining and acid fast staining. هلا why do مثلا TB does not work in gram stain? It's the same reason why it's مثلا إنه it responds to the acid fast stain because of the mycolic acid. تمام؟ وعندنا special staining. طبعا special staining is for if we want يعني if we had a specific microorganism in mind and we know إنه it only works with a special staining. في عندنا capsule staining, flagella staining, endospore staining. From the name we are مثل targeting specific parts of whatever we're trying to see. يعني مثل مثلا هون special staining يعني we're targeting specific properties, structural properties. يعني مثل مثلا الفلاجيلر yeah إنه we're looking for the flagellum. شو كمان يعني that's it, you know. Capsular, we're looking for the capsula. تمام طيب يلا, which one of the following media is commonly used for the cultivation of anaerobic bacteria? يلا guys. C. Okay, perfect. Thioglycolite broth. Perfect. I remember getting a question like in my OSPE. What's the main function of sodium thioglycolate in the medium? So we said anaerobic. So what do we try to do? We try to reduce the agent to remove the oxygen. تمام? Perfect. What is this media called? No, thioglycolate is this one. Something meat. So it's basically Robertson's cooked meat medium, the most random thing ever. So it has meat and yeah. بصراحة just know its name, know how it looks. Maybe يعني they can do a sneaky move and show you that and be like يلا identify. طيب يلا organisms that require increased carbon dioxide concentrations are known as capnophils. Perfect. Because I remember we got a question كمان عن هالشي. طيب هلا هاي the carrier Blair transport medium is specifically designed for no. You are tricked by the way it looks. It's for stool samples. It's written, I think, in the slides, إنه Carrie Blair is used for specifically stool samples. تمام? Or not specifically, but in one of the uses. طيب يلا, let's move on. 29-year-old traveler. She presents with abdominal pain, tenesmus, and bloody mucus-filled stool. And you see this. What is the first thing that you think of? So عندنا نحن watery diarrhea. We see this beautiful thing. What is the microorganism? Think. خفك بس ويطلع الأنسر. Oh, okay. So we have irregularly shaped trophozoite that is ingested red blood cells. صح. لا wait. بلا صح. أنت بهستوليتيكا. You're right. You're right. ما بعرف ليش they did not include giardia in the labs. What is it called? Best giardia looks really special to me صراحة. I really love giardia. Now, these are the cysts of Entamoeba histolytica, okay? How can we differentiate between the kinds of Entamoeba histolytica? By the cysts, the number of cysts. So if we have 4 cysts, that's Entamoeba histolytica. Or Entamoeba dispar. Like it's either this or that. But if we have more than 4, then it's Entamoeba coli. وعلى فكرة ممكن يجيبوا لكم a picture of that and they tell you يلا choose. مثلا يكون فيها 8 and the choices are Entamoeba histolytica, Entamoeba dispar or Entamoeba coli. And you know that it's coli. بس أكيد ما حيحطوا لكم يعني those 2 choices when it's only 4. تمام؟ So the infective stage is basically the cyst. So we know the cyst, if someone ingested the cyst, they're going to be infected. The diagnostic is the trophozoite, which is the thing that we just saw. And what sample do we take to basically diagnose? It's the watery stool. So we take a stool sample. So again, infective is the cyst. So the cyst has to be either in water and anywhere في cyst and you ingested it خلاص infective state it will infect you and then we can see this we can for diagnosis we either use basically التروفوزويتس اللي هون أو cysts تمام and then the patient sample is watery stool and it's fecal oral route so basically most of the time it will be in here, it's called traveler's diarrhea. So patient will be traveling. He is doing whatever. He drinks same water that's contaminated. He eats something that's contaminated. Anything that can have fecal deposits. Okay? Yeah, moving on. What is this very beautiful? I know. Okay. So 35-year-old man, Camp Sudan. He has prolonged fever, weight loss, splenomegaly. Bone marrow shows the following. So we have a mastigote inside macrophages. يعني clearer than هيك ما في. Wait, the notes are gone. Oh, oopsies. I know you guys said leishmania, أكيد. So leishmania, exactly. And leishmania can cause central and cutaneous. So this is cutaneous leishmania. It can literally show you this picture ويحطوا لكم this and a bunch of other مثلا like protozoa and they were like يلا choose. تمام? So know that this amastigote with these like dots that is inside the macrophage, this is leishmania. And it can cause cutaneous lesions, which is this. Now, the infective state is the promastigote. You have to know the name, promastigote, because she might ask you, come on, what is the hypha? You have to know how to write it, promastigote. Diagnostic stage is the amastigote that we just saw. So these are the diagnostic. This is what we see. And where do we take the culture from? It's either going to be from the tissue, إذا كان عنده cutaneous or mucocutaneous, or the bone marrow if it was only visceral أو in the central. تمام? And what is the vector? The vector is this beautiful sandfly. Why did I include a photo? لأنه they might put كمان a photo of different flies and tell you, oh, choose which one is the vector for this. تمام؟ تمام. So sandfly, clear? Is it clear? I hope it's clear. Please تذكروا كيف الساندفلاي شكله. تمام. Okay, يلا next. So 21-year-old student. I don't even have to read it. Plasmodium falciparum. Exactly. Malaria. أصلا the second they say thick and thin blood smears, you guys have to directly think, no, oh, it's something that has to do with malaria. Okay, clear? So banana shaped gametocytes we think of malaria plasma. ما ضروري حتى أقرأ الهاي بس هو يعني usually guys malaria بيكون في chills, fever, non-specific symptoms. So we have here a thick film and a thin film. Now the thick film, we only see dots, 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 dots. These dots are basically the chromatin dots. And على فكرة, again, she might ask you, what are these dots? For what? Come on, chromatin dots from the malaria parasite. Guys, wallahi, يعني I wish I'm exaggerating, but they're like that. They can be very, very, very unpredictable. فإذا we do a thick film, we're trying to just concentrate الهاي, so we see it from afar. So we're not يعني هون we're breaking the RBCs, so you don't see the RBC. Like you see here, we have RBCs and we have this very beautiful ring from the trophocytes. بس هون لا, هون we're crushing the RBCs and we're just seeing how much do we have. Like we want to see the parasitemia, like how heavy is the A. هون thin film, we see these beautiful rings from the trophocytes, and these are like the very characteristic look for Mr. شو اسمه malaria or plasmodium. So infective stage is the sporozoites. Hello. The diagnostic state can either be the trophozoites, schizonts, or gametocytes. And what is the sample that we use? It's the blood. Is everything clear up until now? بس I did not mention something بالإشمانيا إنه بالإشمانيا it's the female sandfly. Guys always know إنه the female is the problem, and females please don't cancel me, but I just mean that for what is it called? No, I mean that only for animals, okay? Specifically mosquitoes. أما نحن الفemales were amazing. Regardless of that, I don't want to be cancelled today. Yeah. So infective stage, sporozoites. Diagnostic stage, trophocytes, gametocytes, or schizonts, والpatient sample blood. And this is our very beautiful vector, which is the female Anopheles mosquito. طيب هلا كيف فينا نفرق بين هاي المosquito والsandfly? Sandfly has these like هيك بتحسوهن like it's like sand particles on it. But this in here, you see how like I don't know, she looks ugly. Sorry, but يعني you see how like she looks evil. Come on. Just use your gut feeling, I think. بس لا, بس sandfly is clearly يعني you can clearly see it. يلا. What is this? Trypanosoma. خلاص. So okay. Motile flagellates with underlaid. So this is the flagellates and it's trypanosoma. Specifically, sorry. ليش هيك؟ اوكي هلا احنا عنا Trypanosoma cruzi and the other one that is Trypanosoma guys, the names are hard. Trypanosoma brucei. So one of them is short and basically what is it called? Short and fat. The الثانية it's slim and شو اسمه? It's slim and tall. I'm literally fat shaming like a microorganism. Regardless of the fact that we're doing that, Trypanosoma brucei is this اللي هي slim and basically tall. تمام. Because this causes what disease؟ اللي هي ال sleep sickness. تمام. And sleep sickness, it's caused by which bug؟ يعني what's the vector؟ It's this tick. لا مو tick, sorry, tsetse fly. صح, you're right. It's this fly. It doesn't look like a fly to me صراحة. It looks really weird. But yeah, you're right. It's the tsetse fly. So again, this is Trypanosoma brucei. It's long, it's thin, it causes sleep sickness disease, African sleep sickness, and the vector is this tsetse fly. This, on the other hand, شوف شايفين, it's actually short and fat. This is Trypanosoma cruzi, okay? And it causes what disease؟ تفضلوا. The floor is yours. No, بالعكس. This caused the Chagas disease. So this one that is short and like fat, right? It causes Chagas disease. And Chagas disease, what is it like? What is it? Exactly, megacolon, and it causes the heart وكل هذا. It will cause Chagas disease, and it is transmitted by what vector? The vector is basically the kissing bug, and we'll see the bug right now. It's ugly. اللي هي reduvet bug. Such an ugly bug. All of them are ugly بصراحة. Anyway, so infective stage is the metacyclic trypomastigotes. What is the diagnostic stage? The trypomastigotes. What is the patient sample? طبعا everything can be a patient sample depending on the symptoms. So blood, because we clearly saw that. CSF if it affects the brain. Lymph nodes. We could do it come from the colon because it causes megacolon. You could do it from the heart, like everywhere. And this is the reduvid, the kissing bug. Unpopular opinion, reduvid looks cool. I don't know. Maybe. I don't see it. يلا Maltese cross guys. خلاص no need to read حتى. Maltese cross is Babesia. Slay. So Babesia and Maltese sporozoite is the infective stage. What is the diagnostic stage? Trophozoite, merozoite. What is the sample? Blood. تمام. And this is the tick. Sorry, Babesia is caused by a tick. So Babesia is caused by a tick, اللي هي الإكزوديس tick. Trypanosoma cruzi is caused by ايش كان اسمها نسيت. No, the tsetse fly. The cruzi tsetse fly and no wait. T cruzi is caused by the reduvid bug. T brucei is caused by the tsetse. Okay? صراحة guys الله يعينكم. God be with you. But I'm sure you're gonna memorize them. يلا let's recap. Let's recap the bugs really fast because I'm pretty sure. If you guys do get a question where she just shows you the bugs, please tell me. Please. Because I know her حركات. So again. انت مين بهستوليتيكا ما في vector, never mind. Leishmania. What is it? Sand fly. It looks like this. Malaria. Anopheles mosquito. She's thin, she's evil. Sandfly has these like sandy-like particles. Trypanosoma. Brucei, which is basically the tall one, the fit one, the مدري شو, caused by the tsetse fly. أصلا look at her. She's like tsetse, like هيك صغنونة. Then Trypanosoma cruzi is caused by the reduvid bug. And it looks like a bug, please. Like a bug looks like a bug. It looks like beetle bug, but it looks like a bug, okay? And then the Maltese Maltese cross Babesia, then it's the eyes with these tick, and it looks like a tick, like, I don't know, it's like tick. It's not like the tsetse. Tsetse is like black and small. Hey, it looks like a spider. A tick looks like a spider. Hidik does not look like a spider. I'm just trying to help you guys remember. I hope I did, yeah. Anyways, yalla. What is this? Egypt. It's a farmer in Egypt. Schistosoma hematobium. صح. وليش hematobium؟ لأنه terminal spine is هيك straight. Now, if we look here, the lateral spine, the spine is going to the lateral side. So it's Masuni, Mansuni. And then this is the ugly mother. Okay. And this ugly mother can go and live in these areas. Wait. This ugly schistosoma can go and live either in the large colon أو بالsmall colon أو بالbladder حسب the kind. So, saying that, you see how we have 3 different kinds. I know, guys, it's not the best HD, but whatever. So this is terminal spine, Mansoni, okay? And then Japonicum, it's like cake circular, and it looks like fluffy Japanese pancakes. Disgusting to even يعني compare, but we gotta do what we gotta do. And then this is the lateral spine, sorry, the terminal spine is like straight, not going to the side. So this is hematobium. Now, hematobium and japonicum can be in the bladder. So that's why we can use urine for, what is it called? For finding these cysts. While on the other hand, Mancini and japonicum, طبعا japonicum جاي بكل محل. Mancini and japonicum, the mother is usually in the intestines, we find the cysts and the feces. تمام؟ Clear? Good? Sick. Hydatid cyst of the liver تجي من الdogs. أو يعني مو بتجي من الdogs. هو basically it's an indirect life cycle بس صح perfect. Echinococcal granulosis. It causes cysts that usually are in the liver. And it looks disgusting. Looks really bad صراحة. Look at her. She looks evil. She looks like a butterfly without wings. مهم. This is a really long worm. And then it has these heads. What is this? Hello guys, I'm just showing you these and I just want you to know them because they're just going to show you. Scolex, صح. This scolex, where is it found on? So عنا ليك وعنا 2 different kinds. عنا this one or this one. This one has the scolex and it's really clear. This one doesn't really have the scolex. So these are the 2 different kinds. عنا Tinea. I gave the answer. عنا Tinea saginata, Tinea solium. So Tinea solium has the scolex. Tinea saginata does not have this scolex. تمام؟ طيب. هلا هدول they're usually in undercooked food. These undercooked food, specifically meat. This meat can either come from pig or cow. طب يلا Tinea solium, pig ولا cow? Pig. وتينيا saginata is the cow. صح. You're right. Now, what is this? A worm. Ugh. Just like a bunch of worms. It looks disgusting. I'm sorry. I think there's one more. Oh. I gave it away. But Ascaris. Now, Ascaris lumbricoides, usually, how does it present? It presents with bowel obstruction. And if you look at the يعني كمية الworms, it can cause bowel obstruction. So it kind of makes sense. And yeah, so we have these 2, these 2 eggs of this. Which one is the infective one? Act like you did not see. Before that, what's the difference between the oval and the round? Cricket noises. So one of them is embryonated and one is not. So this is embryonated and this is not embryonated. Okay? So نحن embryonated is the one that is infective. Okay? Okay. So these bunch of worms are Ascaris lumbricoids, and basically شايفين هاي round little cute one is the embryonated egg, and it's the infective one. تمام. Now let's look at this one. ليش هيك الصورة؟ Wait. No, it's not. Or maybe it is. No, it's not. Exactly. Interbobus vermicularis. بس wait أنا مقهورة ليش الصورة هون ما ظابطة لمين. Fix that. Um, sorry, I can't see. Bring forward, bro. Continue to share. Yeah, okay. So these are the beautiful cysts. They look صراحة أنا من وقت ما كنت a second year, I always saw them as like plate with food on. I don't know if I was always hungry while studying, but يعني it really looks like a plate of مثلا riz or something like that. Like I don't know, I tried to gaslight myself into seeing something that makes me remember it and associate it with different things. بصراحة that's how I survived. Plano convex. Wallahi, that's how I survived. Like in POD, wallahi, خصوصي with these images, you have to literally gaslight yourself into thinking you're seeing stuff, even if you're not seeing them. يعني أنا هون مثلا, I used to remember, but the thing is, there's another one that kind of بتخرب الدنيا, but I found a way to help يعني. اللي هم هاد صحن. Actually, you know what does it look like? It looks like mahar. You know the mahar, which is basically, what is it called? Oysters. Oh, حتى the other one. Yeah, so basically it's like you open an oyster and it looks like that from the inside. Like, even if it doesn't, guys, just ignore, gaslight yourself thinking it does. And the very, very, very characteristic thing about this is basically, the worm at night, exactly, the worm at night goes, and where does it go? It goes into the person's butt, and then it goes out. So when you do the Scotch tape test, you can see everything. Pretty disgusting. Interbose, very reckless. Whatever. لا مش هيك ما اتفقنا هيك. Anyways, ورا guys في no, we have to see it صراحة. We ruined that. You guys know what it is now. But like, oopsies. Let's ignore. Wait, شو عم بصير? I'm sorry. Oh. They don't want you to see the first one. Ah, I know the problem. Content problem. Okay, now it will work. So you know the answer, but regardless, let's just act it out. Continue to share. So you see this, guys, when I looked at this for the first time, I felt like it's, this measure tape? You know the measure tape? Well, it looks like a measure tape. Because it's like, I don't know, it looks weird. بس يعني شايفين woo, like a measure tape. Anyways, regardless, this is how it looks when we look under like something, and this I wouldn't have guessed, honestly. Okay. Okay, hear me out. مشكلة it's really sad, بس يعني bipolar plugs. Oh, I like it. But it looks like a tray that you hold like this, okay? And then in the tray, to help you remember the name, there is tiramisu, because it's trikuras, like I don't know. That's just the way I memorize stuff. So trikura. So it looks like صينية, It's like the thing that you serve food in. The other ones looked like an oyster when you open it. So let's go back. I hope everyone sees that. Bottom. This is يعني صراحة this is like an alien يعني if you don't know Tinea solium and Tinea saginata, that's your problem. They're aliens. But this خلاص كمان but this is an oyster. So this is what Interbos vermicularis. The صينية is the Trichuris tricura. تمام؟ Okay, I know the problem. Okay, wait, guys. There was a mistake from my end. This is not here. This is not here. And that's why it did not appear. This should not be here because this is not this. This is not Trichuris trichura. Trichuris trichura is only this and this. Okay, ignore the one that we just saw because this is something totally different. Totally. So what do you guys think is this? So حكينا interbius vermicularis. حكينا عن ما بعرف شو. You're right. No, you're not. بلا, you're right. It's intestinal. أنا قررت أتفزلك. بدل ما أكتب intestinal hookworms, I put ankylostoma. Ankylostoma. But why did I choose this photo? Because I remember, I don't know if she removed it. I don't know if it's in the lecture, but I remember a clear photo of this face. Like microscopically, but I couldn't find it. But هلا, the idea of this, these hook up into your intestine and your bowels. And that's why they have this like claw-like shape. So let's go back and look at it. It's not clear here, but in here they have something that will help them to catch into your walls. Just stay there, sadly. So let's try find something to describe this so we remember it. I can't think of anything. So هديك oyster and the tiramisu tray. But what is that? Sorry guys, I can't think of anything that can help you. خلاص, ankylostoma is the thing that is out of the blue. Is that clear? I think it's clear guys. يعني we went through all the photos. We explained. هلا let's complete يعني قبل ما نكمل. Let's revise مثلا إنه revise these things together. يلا. So schistosoma. نحنا we know إنه schistosoma it's indirect. تمام. So the cycle is indirect. الhost نحنا we are the definitive host. It can be found in snails, but sometimes the scenario would be like someone who eats snails. Because yeah, the French do that. Good for them, I guess. Or someone who basically swam in water. This is the most يعني هاي. So they swam in water. It penetrated their skin. What is the infective stage? The cercaria, which is basically a worm itself. It will like go somewhere. And then the diagnosis stage is the eggs that we saw. The eggs that we saw are really, really like diagnostic. يعني أصلا انتوا guys كمان you would know them from the spine. If it's lateral, if it's like a terminal spine, هيك it's the hematobiquum, whatever the name is. Lateral, it's Mancini, and if it's circle, it's Japonicum, like a fluffy Japanese pancake. مصرة. Tinea, حالنا tinea solium, tinea sanguinata, كمان they're indirect. We are the definitive host للأسف الشديد. Intermediate hosts are basically pigs and cows. Or cattle, not specifically cat house. We can كمان be intermediate hosts. طبعا on all of these there's no vector. كلهم. they're not like protozoa. They have a vector. No. They just directly come and invade us للأسف الشديد. So tinea, it's by the ingestion of undercooked or infected meat. طبعا undercooked meat بده يكون فيه cysts. تمام؟ الانفكتيف stage عنا oncospheres. I don't want to go into the details. I don't care. خلاص, infective stage is the eggs. إذا نحنا we ate, إذا نحنا هدول spheres are جوات undercooked meat, they infect us. Just know the names, guys. And the diagnostic stage is basically whatever we saw. اللي هي you saw them, the proglottids. They look weird. Echinococcus is indirect كمان. Definitive host is the dog. And we are accidental hosts بالصدفة. How is it transmitted to us? It's transmitted to us through fecal oral. So basically, anyway, where if we ingest anything that is contaminated, we can get echinococcus. Infective stage is the eggs that are basically in the contaminated bliss, and then we could see the cysts. Do you remember the cysts? The cysts usually happen in the liver. Next. Ascaris lumbricoids كمان نحن ال definitive host ما في أي شيء. fecal oral eggs كلهم أصلا شايفين eggs. Enteroboas vermicularis كمان fecal. Fomites, do you remember guys what fomites is? Fomites is basically when you're just like dusting something and it goes and then you can inhale it and then sad for you صراحة. My favorite honestly is the ankylostoma because it's skin penetration. يعني you would be usually it's somewhere where you're barefooted walking on a sand and then it يعني holds into your feet and then finds its way into your body يعني slay queen for her صراحة. But the rest are boring. The rest are all fecal oral. تمام? Clear? Except for the pinworm اللي هي Enteroboas vermicularis. Why are we saying fomites? Because basically when a baby sleeps and the worm decides to leave out and go, it might leave things in the bud يعني of the baby. Now let's talk about this very beautiful guys. I love fungi. I don't know why, but I love them. Now, what is the stain that is used? It's blue. Lactophenol blue. Perfect. Now, lactophenol blue. Now, why is it important? طبعا guys أنا ما حفظت هالديتيلز because يعني trust me ما ححفظهم بس it contains phenol. Okay. Now the phenol will act as a disinfectant. It will kill any living organism. The lactic acid is there to preserve fungal structures and the blue is just a color. So the idea is lactophenol, the phenol is to kill anything else, but lactic acid is to preserve it. I don't think you need to know this. Now let's look at this. What are we seeing in here? In here, we're seeing budding yeast cells. What are the budding yeast? There's these. شايفين عنا 1, 2. They're budding. Buddies together, budding yeast cells. تمام? They are connected لشو؟ a pseudo-hyphae. Now, what is a pseudo-hyphae? Why do we call it a pseudo-hyphae? هلا hyphae, when we say hyphenated or something, it's like the stem. You know how we have like trees and they have stems and whatever. So it's like a stem, like an extension of a stem. حلو؟ هلا this extension of the stem in here, wait, I'm not ما بعرف كيف اشرح لكم اياها. Let's explain. Okay, wait. This is a let's think انه هاد a true hyphae. Now a true hyphae, it's like connected all together. It's like a stem. Pseudo hyphae, it's basically multiple cells that are combined together to make it seem like it's a hyphae. So like if you closely look, it's like 1 and then 2 and they're like شايفين أكتر من واحدة. They're not fully connected together. They're just cells that come in the shape of a hyphae. So this is a pseudo hyphae. Is it clear? Okay. Now, this organism is the same as this organism. Now, what is the agar that we use in here? It's the SDA, which is subord agar, which is used for fungi usually. Tamam? Tamam. Now, it looks like pearls. It looks like classy, beautiful, nice. But in reality, it's not. It causes مثلا white plaques on your tongue. It causes طبعا it does not cause this on يعني normal, not as a normal يعني if you have a healthy immune system, it's not gonna Candida is not gonna infect you. It usually infects immunocompromised patients. All right. So what is this? Candida, perfect. I love Candida. Now, what's the name of this test that we use to further check and know it's Candida? So what do? We take it, we apply 0.5 milliliter of serum that has glucose, and then we keep it in the incubator for a couple of hours and see. What is the test called? Germ tube test, exactly. So we were going to see if we're going to have these germ tubes that will grow after adding these glucose to it. تمام? This is like a confirmatory test for Candida. Now let's look at this beautiful, beautiful structure. I love it. Wallahi guys, fungi are so beautiful. So let's describe. أول شيء in here, this is hypha. This is شايفين هون? This is a true hypha. It's not like multiple cells connected. It might be segmented segmented يعني there are segments but it's truly connected together. And then we have هيك flower shaped and we have multiple conida, I don't know, but these spores, okay? They have a specific name with a C. هيك كلهم هون together, and then when we look at it like crossly, it has a velvety appearance, then the center is black. What is this? صراحة I love this. It's aspergillus, okay? And guys, oh my god, I feel like a big bag because everything I compare to food, but it looks like aspergillus. Don't ask. Sorry, asparagus. So كنت أنا دايما بحط حافظة بمخي asparagus, aspergillus. Asparagus, aspergillus. But it kind of does look like an asparagus. يا ريتني حطيت صورة an asparagus. Do you guys see it or do you not see it? Because if you don't see it, I'm gonna literally pull up an asparagus and show you. Do you guys know what an asparagus is? Yes, that's how I remember it too. Okay, I'm not a big bug, alhamdulillah. Unless we're both big bugs. Look! Looks just like an asparagus. Anyways. Moving on. In here. شايفين ما أحلى هالhyphen. This is what we mean by hyphen. This is a real one, not a pseudo one, because it's like هيك come on, it's non-segmented, like هيك, it's just like ding, perfect. So it looks like, you know, the dandelions. So how like إنه dandelion بتكون هيك بعدين بتنفخيها and it goes. It looks like a dandelion to me. In here it looks like a flower, like sorry. In here it looks like an actual flower, but in here it looks like a dandelion because it's like that. But when you مثلا look closely, it's like a bunch of small things that make it all together. Now, if we look at it grossly, it looks like a cotton. Cotton. حتى أصلا if you leave it for a while, it looks like mold, and it is mold. يعني it looks like the mold that's on bread. And basically, if you leave it for long, it will change color to brown. So this is the mucor. تمام؟ هلا last but not least عنا immunology اللي هي basically it was your last lab. قبل ما أفوت فيها, is everything clear up until now? Did you guys have like a rapid revision? Did you يعني will you remember any of the things that we talked about today? Hopefully. I know there's like a couple of people. Actually, it's only one person. صراحة, thank you for the person who's attending. But I know people will watch the recording. Hopefully. تمام. Now let's look. Okay. This is a beautiful idea of أنا ما بعرف ليش حطيت هون إلايزا قبل ما أنا بلش حتى. Wait. ما بدي أحكي عن هذاك. Let's ignore that slide. Let's go to the slide. هلا أنا when I looked at the slide, I was like, what? I was so confused, guys. I was so confused because عن جد يعني it looks really confusing and it's so weird. But when I read it more, فهمت عليهم وتذكرت إنه أنا أصلاً I took this and it was really easy. The whole idea is أولاً this test is called this name. I don't even know how to spell it. Ouch, Terloni. It's a big ouch. Anyways, so the idea is from this test, and I don't know how do they actually see the يعني how do they يعني I don't understand the gain out of it, but you need to understand the mechanism. So the mechanism is إنه هن they put يعني مثلا هون we have a material. Let's say هون عنا an antibody وهون عنا an antigen. حلو؟ هلا this antibody and antigen they're not ملزوقين ببعض. They're on space. And they allowed them to diffuse. So they're going to diffuse. طبعا they diffuse in an equimolar يعني إنه مهم إنه in a way which is equal. So they diffuse, and then when they meet, they react. And when they react, they cause the formation of this white color. Now, this white هو basically precipitation of the reaction. This white is in a distance that shows equal amount from both sides. يعني ممكن مثلا by the way ممكن the concentration أنا اللي حاططها هون للantibody. Okay. ممكن the concentration in here is not as like how can I explain it? It's not as okay. So basically maybe هون حاطين a diluted. So maybe sometimes let's draw. So random. But let me explain. I have an idea I want to explain and I hope you guys understand it. So sometimes it would look like, مثلا, let's say this is a circle, this is an antibody, and this is an antigen. حلو. But the line, let's say, is not supposed to always be in the middle. Why? Because this might be هاي الانتيجين. Okay. Anti B. وهاي الانتيبودي anti B. Okay. So why is it not at an equal stage? Because the antibody in here might be at a diluted amount. So the amount that is needed to form this reaction is يعني بدها more space وبدها more particles to travel. هون ما بدها a lot of particles to travel because it's at a higher dilution. Do you get what I mean? Is it clear? But just understand. No, it's not a rule. It's not a rule. إنه antigen diffuses slower than antibody. It's just what I'm trying to say is إنه يعني مثلا شوفوا هون this is, let's say this is the antigen. حلو. Let's say, oh, this is an antibody. Well, I don't know what this is. Whatever it is. Okay. And this is an antibody. And this is an antigen. And this is another antigen. هلا ليش هون it's closer than here? Like why did this have to travel a longer distance than this? While in here مثلا it's kind of equal. What I think it is, في عنا different dilutions. Do you know what I mean by dilutions? يعني هن مثلا when they prepared this, they put, let's say هون 50 milligrams of this antibody. And then they put here antigens. Let's say here مثلا these two antigens work with this antibody. بس مثلا the rest, they don't. They're not related. They're doing a reaction. So مثلا let's say this was مثلا 5 and this was 10. فلأنه مثلا this was 10, it did not need a lot of distance to travel, it did not need a lot of particles to travel. بس هون لأنه it's 5, it might need more particles to travel. المهم الزبدة اللي أنا عم بحاول أوصلكم إياها إنه the line is not always at the middle. It depends on the concentration of the different antigens and antibodies. تمام؟ بس لأنه I don't want you guys to مثلا let's say اوكي هلا انا كيف بيعمل بلان ما بدي اياكم later on مثلا you see it and then it's not in the middle and you think it's not Arch-Teleroni or whatever this thing is. You see يعني هي هاي الفكرة. I think it's clear. How do they use it? I don't know. And I don't think you guys should know. I don't think you care. I don't think I care. I don't think anyone cares about this. With all my respect and love to Dr. Garman. Next, this is basically radial immunodiffusion. هلا هديك كانت الouch I don't know what the name is. It was double immunodiffusion, as in like the antibody is diffusing towards the antigen at a place where basically both of them are equal and they precipitate wherever they meet. هون, it's radial immunodiffusion. So basically, let's look. So basically we have, let's say, this is, let's say, the antigen. Okay. Wait 1 second. Okay. So this is the winner. Sorry, I got distracted. Okay. شايفين the green thing? The green thing is basically the antibody with the agar. And then we put this circular thing. We put these dots, these dots, these dots, these dots. These dots are basically the antigen. Now, what we are trying to do, we're letting the antigen, هن هدول circles, they're called wells. So they're هيك انه empty spaces where you can put fluids. Okay, نحنا كله هذا guys experimental. It's يعني they do it for immunotherapies and stuff, like to experiment stuff. So they put like, let's say 2 drops drops everywhere. حلو. بعد ما يحطوا the drops بيشوفوا, they measure the basically the radius of the circle around, because that's where basically شايفين هون الرم, this is where مثلا the precipitation reaction happened. So this is basically equivalent ل the antigen concentration. It's all يعني مع كل احترامي للإميونو, it's all like, I don't know, nothing concrete, بس what is it called? It's just something you need to know. You just need to know the concept. The concept is, this is used to check antigen concentration. How do we know the antigen concentration? From the ring diameter. Clear? I hope I'm not confusing you guys even more, or something. Yes, so the agar itself has the antibodies. Yep. The agar is mixed with the antibodies, and then we add tick the antigen, and then the diffusion is the antigen concentration. So نحن إذا عم نعمل إذا نحن عم نعمل شو اسمه we're doing an experiment and we want to double check that, we can do that, okay? Is that clear? Okay. Now ELISA في عنا so many different what is it called? So many different things. But let's talk about wait. هلا نحن we need to كمان make sure that we're understanding and all of these different things. What is happening? The baseline of the idea is we have an antibody and an antigen and they are reacting together. حلو؟ هلا نحن هون عنا something called direct ELISA وعنا something called indirect وعنا sandwich ELISA. The main idea is indirect. Let's talk about direct before indirect. The direct is basically the photo that I had in the beginning. So this is latex agglutation. My bad. Okay, هلا بنرجع للlatex. بس let's talk about ELISA. The main concept of ELISA is, and let's talk about direct ELISA. We just have this well, and in this well I put an antigen, and then I get an antibody من برا, and I want it to bind here. Okay? هلا indirect ELISA, once they bind بصير في عنا a color change or something. تمام؟ In indirect ELISA اللي هو هون, what happens is we're going to add an antigen. حلو? This is the antigen. Then we're going to add a primary antibody, like an antibody that recognizes this antigen. فنحن عنا هون a complex. Antigen antibody complex. تمام؟ What we're going to do is we're going to wash. So we're going to remove all the excess and we're only going to have the complex that we have right here stick in here. حلو? Then we're going to get an antibody that recognizes the primary antibody. So this antibody in here will recognize the antibody that is already attached to the antigen, which is already the complex. So in other words, the antibody is going to identify the complex, and then a reaction will happen. مثلا ممكن يكون a change in color, ممكن يكون precipitation, depending on the whatever experiment they're doing. Is that clear? Okay. هلا عنا another thing which is كمان another type of ELISA, which is the sandwich ELISA, which is the ELISA, by the way, that is used in pregnancy tests. The idea is we're not going to put antigens in here, but we would rather put antibodies. So we are putting هون عنا هيك, عنا هون antibodies, and then we'll add the antigen. So this antigen is going to bind to the antibody, and then we're going to wash it. So all the excess is gone, and we have complexes right here. And then we're going to add another antibody that will basically bind. We will add an antibody that will bind to the same antigen that is already binded to the primary antibody, they will all form a complex, and this complex will basically cause a change in either color, precipitation, etc. هلا مثلا in pregnancy, that's مثلا نحن what is the thing that can tell us someone is pregnant? Basically beta HCG. تمام? This is a hormone. This is an extra information, guys. I'm just trying to help you understand the idea. So نحن بنحط هون something. So this is the pregnancy test, the one that the women usually pee on. So what does it have? It usually has these antibodies, all right? Oh my God, okay. It has these antibodies that basically will detect, let's say, beta HCG. So نحن عنا beta HCG. So basically they peed, all right? Wait, no, it does not have beta HCG, بالعكس. So what happens, wait, never mind, this is wrong. So what happens is we have antibodies. هي هاي الفكرة. عنا antibodies هون. And these antibodies, all right, they're already connected to something. They're already connected to an antigen. So something that will detect this hormone. حلو? So once the woman pees on it, okay, بصير في عنا an interaction between this hormone and this antigen, which will bind to it, and that's the reason that the color changes on the strip. Is it clear? يعني this is the idea. إن إحنا عنا an antibody, and then the antibody is connected to an antigen, and then this antigen would either مثلا react with beta HCG or the hormone, or would react with an antibody that is found specifically in pregnant ladies' urine, and then they will form a change in color, and this is what the sandwich ELISA is. Did you guys understand the fundamental؟ يعني أنا بيهمني إنه أنتوا فهمانين the idea. I don't care about the mechanism, just the idea. إنه نحن عنا it's either مثلا an antibody, so it's either we put antibodies. Exactly. We put antibodies and the antigen is sandwiched between it. أو بنحط الانتيجين and we expose it to antibodies, and the second antibody that binds to the مثلا initial antibody is the one that's مثلا fluorescent, okay, or is the one that reacts. يعني that's the idea. It's just a bunch of antibodies. And هلا في عنا something called quantitative ELISA. Now quantitative ELISA is basically an ELISA that helps us know the quantity of something. So if نحنا مثلا عنا an experiment, and this guys is done in experiments a lot. So if we want to know the quantity that is effective مثلا, or the least amount that can cause a reaction. So شايفين كيف بيعملوا مثلا, this is they do a bunch of dilutions. So this is, let's say, different things. So we want to know مثلا the concentration of something. عنا A, B, and C. What do you think it has the most concentration? Obviously, it's going to be C. Why? Because شوفوا كم dilution it went through لحتى the color changed. Do you get what I mean? يعني نحن عنا هون a reaction happened, بس نحن we want to know مثلا the concentration of the antibody that we put or the antigen that we put for a specific مثلا exam. So basically we're going to keep on diluting, and then once the color becomes more and more normal, this means إنه خلاص it's getting diluted, يعني the high is gone. So we can kind of know a little bit how much is the antibodies or high. فهون مثلا in A the highest was 116, okay. إنه يعني هذا التايتر كان the highest where the color changed, يعني highest titer as in like this is the maximum titration that was reached before the color went away. B it was 1-2. C it was 132. So obviously this is more concentrated because it was able to go through more titrations. تمام? Quantitative ELISA كمان is used in research. Now, flow cytometry and ELISpot, guys, these are things I don't think you really need to know. Just know the idea. هلا flow cytometry, it's basically a machine. In this machine, they allow guys, this is all research, by the way. يعني there's literally minimal يعني انتوا as I don't think you're gonna see يعني if you're lucky to do مثلا research internships outside, or if you do research هون. يعني مثلا I saw a flow cytometry, I've done ELISA, I've done all of these things because I went out on a like research internship وهيك. فإن شاء الله يا رب انتوا كمان تقدروا تطلعوا. بس all of this is just literally research. يعني it's not clinical, you know, like it's not something that would help in مثلا immunotherapy and stuff, but other than that it's not going to help a lot. Just know an idea, because يعني obviously as a physician you have to know everything, not everything, but إنه يعني have knowledge about most of the things, that no need to have deep information about it, but just know about it. So the whole idea of flow cytometry, ما أعرف ليش يعني هاي الفقرة ليش كانت مهمة. المهم the whole idea of flow cytometry is that we are trying to measure the amount of T cell subsets. So عندنا نحن, we know we have two kinds of T cells. We have CD4, CD8. We want to know مثلا in this specific sample, let's say نحن we are doing a research على some patient, and this patient we think he's immunocompromised, or we think there's something wrong with his T cells, or we think مثلا we gave him a medication and we want to see if it activated the T cells or it did not, whatever reason. So نحن we want to count these. So what happens is it's a big machine, you put your sample in it, it's going to allow, let's see it هون شايفين, it's going to cause cells to go هيك إنه شايفين in row by row, and then the flow cytometry is going to put a laser beam, and this laser beam is going to count مثلا this is CD8, this is CD4, this is ما بعرف شو, this is ما بعرف شو, and then it's going to keep account of the cells. Okay, nothing يعني you need to know, guys, nothing we can do, you know. And then the ELISpot assay, let's look at it. 1 second, بس let me erase all icon slides. Okay. ELISpot, it's like ELISA, but each spot will have basically a specific مثلا cytokine released. Okay, so let's look in here. So what do they do in here? They put, let's see. Okay, عنا هون هدول شايفينهم antibodies. حلو. This antibodies بيحطوهن مع a specific cell that will release specific antigens. يعني شايفين مثلا this cell, let's say this is a macrophage and it's releasing مثلا interferon gamma. Okay, then you're going to remove the cell and then you're going to try to add basically an antigen مثلا new created to see if it's going to bind to the interferon gamma ولا لا. And then after you incubate, okay, after you incubate, all right, guys, this is not important. Just the whole idea is, you know, we're trying to count the amount of active immune cells by مثلا let's say شو اسمه. So let's say مثلا, I'm trying to think of something that can يعني make sense. Okay, let's say نحن عنا okay, that makes sense. We have a white blood cell and we got a bacteria. We want this white blood cell to act against this bacteria, and we want to count how many immune cells are done. فنحن we already charge مثلا, we charge this, شايفين هاي مثلا macrophage, we already exposed it to the bacteria. So هدول اللي هون they're supposed to be against the bacteria but they're not against the bacteria. So بنحطهم هون. And then these antigens are binded to the antibodies. And then we want to count how many antigens were formed. So we send in something that would bind to this antigen. But هلا نحن when we send something that would bind to the antigen, let's say نحن حطينا, let's say 10 particles, but in reality only 3 binded to it. So what would we do? We would add a third one that would just bind to the antigen اللي نحن we already sent, the antibody, sorry, that we already sent to bind to the complex to know how many. So let's say مثلا I sent in 10 but only 3 binded. I will send the next day something that is specific to the kind of whatever I put. هاي شايفين هاي مثلا, something that's specific to this just to count how many cells are there. This specific thing يعني مثلا I sent in let's say 10 yesterday, I'll send 10 again today in hopes of مثلا let's say we have 10 cells, but in reality we only have 3. So these 3 will bind to that, and مثلا we will only have 3 مثلا light, let's say إنه هدول in wells. فبصير شايفين هون color product. So the color product is only when these bind to it. So we're going to have مثلا 3 spots, and these 3 spots indicate إنه إحنا عنا 3 active immune cells. I hope it's clear. I hope إني ما جبت العيد وخربطكم. Is it clear? Okay, good. Good. Next. This is from the pharma. Guys, did you go to the pharma lab? Like, is this little pharma lab still exist? So, نحن هون, we're doing an experiment, okay? We are trying to see if basically عنا agonist وعنا antagonist, تمام? And we want to see the response of the smooth muscles, تمام? So we added an ascending dose of acetylcholine, so we're increasing the dose of the acetylcholine that we're adding. And then we added كمان histamine, which is an antagonist. And we wanted to see if the smooth muscle would react to this antagonizing thing, and it would react to increase in dose. تمام? So let's start. Did the curve shift to the right? Yes, it did. Did the maximum response change? Yes, the maximum response. No. Sorry, never mind. The maximum response did not change much because like شوفوا مثلا هون, بس it did kinda يعني شوي. بس إنه it was shifted to the right. Does the graph prove that histamine is a competitive antagonist to ACH? I think it does. Why? لأنه نحن after we added an antagonist, the reaction to happen took more time. هلا the maximum response يعني let's say the maximum response did not really change. يعني a maximum response إذا كان it would change, it would probably be something like that. فيعني it's I know some people will argue and be like, yeah, it did change, but it's not like a great change. ف is histamine an antagonist to acetylcholine? Yes. طيب. What about atropine? What do you think? How do you think the graph would react if we added atropine? Now, atropine, is it an agonist or an antagonist? What is it? Okay, so if we added atropine, what do you think would happen? Because atropine, is it competitive or basically maximal response decreases? Exactly. Why would the maximal response decrease? هي نحن الفكرة اللي عم نحاول we wanna address is it's being competitive or non-competitive. هلا competitive means إنه it works on the same site as the main drug. So نحن acetylcholine and histamine they act on the same site. So they're competing over the same place. بس مثلا atropine it's not a competitive what is it called? It's not a competitive antagonist, but rather it's a non-competitive. So it does not care. It's not gonna fight it over its place because it has its own place. But it's just gonna go and antagonize it, and the maximal response will decrease. ليش؟ لأنه it's not fighting over the spot. خلاص, I have my own spot, and I'm just gonna decrease your action. Lol. You know? Do you get the idea? Okay, slay. Let's move on. So these are but some photos of things I did not include, but I think they're worth mentioning. So what is this? This and this. You know, what's the first organism that comes into your mind when you see this? Pseudomonas aeruginosa. Exactly. It's green, guys. Green. إذا إحنا شفنا anything that's green, it's Pseudomonas aeruginosa. And I've not seen any other agar that's green. Do you guys agree? I think I agree with myself, صراحة. Anyways, next. This, what do you guys think is this? So this is basically, yeah, which is basically, this is Salmonella Shigella agar, and we have Salmonella on it, and you can see how it's black because of the formation of H2S. And how is the H2S formed? It's formed because the medium has ferrous sulfide, and then the ferrous sulfide will react with salmonella and بيصير في عنا إنه salmonella will release. So what is the virulence factor that causes H2S to form? Do you guys know? So basically, salmonella is going to release a virulence factor that would react with ferrous sulfide, resulting in the formation of H2S, which is this black thing. فإذا شفتوا this, no, it's salmonella. إذا شفتوا this, no, it's pseudomonas. That's it. Thank you everyone for watching. I hope I made it clear. I hope I helped you guys revise. And any questions, you have my email, you have my number, even if it's not on this PowerPoint. I'm pretty sure you guys have it from previous PowerPoints. In the last slide, both green ones are. Yeah, both of them. That's it. Have a beautiful day, and I wish you all the best. الله يوفقكم يا رب. وإن شاء الله you get the grade that you guys want and deserve and hope for. وإن شاء الله it was going to be easy. If you have any questions, my email, number, WhatsApp, everything is open for you guys. شكرا.